twin. If it was your twin, there's no problem. They probably would have exactly the same 26 letters. But what's the probability that someone else out there might have those 13 pairs of letters? And for this, the police and the forensic technicians have what they call a probability factor. And they go through a long statistical analysis, and then they come to the courtroom and they say, the chances that some random person out there matches this is 1 out of 10 billion. They come up with a figure. Now, some of the scientists have debated those figures. It's not clear-cut. There are debates about those figures. It's still not going to be a small number, but it may not be as large a number as 1 in 10 billion or even 1 in a billion. And the closer you are to the family or to the inbreeding of the individual, the greater the chances are that you're going to get a random hit that would match those DNA profiles. So it is an estimate based upon probability factors and certain assumptions. And in order to do this, you need a sample of the population. Let's suppose one of those numbers is 3 and 5. One of those pairs is 3 and 5. You have a sample of the population. You have to ask, how many people in this sample of the population would have a 3 and 5 repeat at that particular part of the chromosome? Let's suppose it's 10%. Okay, then you have to do the same thing for each one of those sites. And, but then you have to ask, is that sample of the population a random sample of the population? No way. They don't have a random sample of the Earth's population. So they choose a population and use that as their, as the sample that they use to determine what the probabilities are for each of those sites. Anything more would go into more complex statistics. Contamination, however, is the most probable cause for mistakes. And also, there's one other area. Mixtures. A lot of times the DNA comes in mixtures, and you have to sort out. You have to sort out, well, let's see, is this a 3-5 or a 7-9? Or is this a 3-9 or a 7-5? Do you see what I mean? You've got to get 2 numbers for each individual, and if they're mixed, you have to figure out whether the mixture, what are the numbers for that mixture? And there are mistakes made, or there are presuppositions made about the mixtures. And unless the defense attorney knows this, they can't challenge the prosecutor that the interpretation of the mixture is an interpretation. That's another area where fallibility comes in. Another area where fallibility comes in is sometimes the DNA samples are not complete. They don't have all 13 pairs. Maybe they have 12 or 11 pairs. In that case, they have to project into the 13. They'll say, well, we couldn't get 13, we only have 11. Well, 11 pairs, you're going to get a lot more people in society with the same 11 pairs, much more than you would with the 13. So those are the areas where there's interpretations going on, mistakes, and of course, if the police know their suspect, they have in their mind, they want that suspect. So the best thing to do is to isolate the people who do the analysis from the people who do the police investigation. And in some countries, they don't allow the police and the police agency to do the forensic DNA testing. They have a completely separate division that does that because they know that once there is interpretive possibilities, that the police are likely to say, boy, that's the guy, we know that's the person. Yes, We even have a way of interpreting the mixture so that person is guilty. And that's not a good way to do science. So those are some of the areas. Yes?
I was wondering, given how sensitive these DNA testing techniques are in amplifying, how easy is it for someone to surreptitiously steal your DNA and actually intentionally plant it?
Oh, that's so true. As a matter of fact, prisoners know more about DNA than the general public. They have to know about it because, first of all, some of them are innocent. And we've had over 250 people in prison who are exonerated because their DNA didn't match the DNA in the crime scene. But one of the things that prisoners talk about is how to implicate somebody else. Just throw something on the crime scene with somebody else's DNA and the police are going to be at it. So if you're in a Starbucks and you drop your cup in the waste paper basket and you leave, and there's a crime, the police, and then, the person behind the counter says, I think the person had coffee here before they left. And the police look where the light is. They look in the waste paper basket. They pick up the DNA. If your DNA is in the database, well, you're going to get a knock at 3 o'clock in the morning. You were in Starbucks at that time. We want to question you, even though you're perfectly innocent. So, yeah, you're going to get DNA at a crime scene, perhaps, that nothing to do with the crime. And that could be planted there very easily, because it just takes a minute amount of it. Because the techniques for copying DNA have gotten so sophisticated, PCR techniques, like I said, I put my thumbprint on there, they swab it, there are few cells. They can use a Xerox process to replicate the DNA, so they got enough of it to be able to actually test for the 13 pairs of numbers. Because it's so sensitive, the likelihood of contamination gets greater and greater. And the story, the first part of the book, is about a serial killer in Germany. They were looking for a serial killer for 15 years in Germany, and it was a woman. Every murder scene, her DNA appeared, and they spent millions of dollars trying to find the serial killer, and she eluded them. And it was embarrassing for the police, not as embarrassing as what they found out eventually. Finally, this serial killer female DNA appeared in a place that even the greatest advocates of the theory had to agree was very unusual and very unlikely. So they figured out something else. They went to the factory where they made the swabs, and they found this little old lady making the swabs and contaminating the swabs. It was her DNA that appeared on all of these sites. And of course, the German police were terribly embarrassed, and enormous cost of money to track this serial killer. And it was not a serial killer. These were not one person who committed all these crimes. But you can see the contamination could come from many different sources. And one of the major sources is from police who are handling the evidence. There's been a lot of discussion about and evidence that police contaminate their own samples. So these are some of the problems with placing DNA at such a high level. Some countries will not allow a prosecutor to convict a person strictly on DNA without any other physical evidence. That's how strongly they are, that you just can't depend upon DNA by itself. And yet Leiterman was convicted just on DNA. They had nothing else connecting him to this young woman 35 years before. Well, this is an interdisciplinary graduate program in public policy, which historically has been named Urban and Environmental Policy and Planning, but it contains a lot of people with, who address different issues. I happen to address a lot of science policy issues. So it incorporates, though, city planning, it incorporates housing, it incorporates child and families. And well, I started studying physics, and then I started asking questions that my professors were not interested in answering. And then I knew I had to go into something deeper, the philosophy of science. So I then started to turn my studies away from physics per se to philosophy of physics and philosophy of science. And that eventually got me into science policy issues. And from there, I just began looking at challenging questions and following my intuitions. Well, each issue I choose sort of gets me out of my comfort zone. And probably the last issue, the one I recently got involved with, which was criminal justice and forensic DNA, had a lot of challenges associated with it. I had to learn everything I needed to know about forensic science, and I had to study the law in a way I've never done before. So it was quite challenging to be able to combine many of these disciplines, sociology, forensic science, law, into one comprehensive book. There hasn't been a lot of resistance yet to the book on genetic justice. And if it were to come, it probably would come from the criminal justice sector, and they usually remain pretty quiet. But in other books I've had that dealt with ethics and medicine, I've had a lot of resistance and a lot of criticism for people whose interests, whose financial interests, were going to be impacted by the book. And of course, whenever I write about chemicals and health, the chemical industry is always watching me. Well, every time I get involved in a project, it becomes an obsession. You almost have to do this when you're probing deeply into subject matter. But I try to balance my life with family and friends, and I live both in Cambridge and in New York City, so it provides enough of a diversity of activities. But I'm very focused when I work on a subject, not unlike somebody who's making a film and blocks out everything else that they do while they're doing it. I think that people in academia like to see their ideas get out to the largest audience possible. At the same time, they usually publish in publications that are viewed as, appropriate for their discipline. And it's a tough balance. So most of my books are from academic presses, and it's not a popular press. But these academic presses sometimes have mass media markets, and that's what I try to write in for people who can take the take the effort to read carefully a book that might not be a popular memoir or something like that. And I always try to explain the science as well as I can to people who haven't had the background.